press the bell icon on the YouTube app and never miss another update.
Hello everyone. So this is my second video on the chapter Cell, the Unit of Life. So in this video we are going to understand the ultrastructure of plant and animal cell, and along with that we will study the structure of plasma membrane. So today's video, ultrastructure of plant and animal cell, which is on the board. I hope everybody can see this ultrastructure of plant and animal cell. So as revealed by the electron microscope, so these two are the eukaryotic cells. So we have already understood what are prokaryotic cells and eukaryotic cells in the earlier video. So the prokaryotic cells are the cells with the primitive form of nucleus or an incipient nucleus, or with nucleoid where the nucleus is not covered by a nuclear membrane. Here the eukaryotic cells, in both the cases you can see that the nuclear material is surrounded by a very definite nuclear membrane. Thereby you can conclude that these are eukaryotic cells, and along with that you can see so many organelles which are embedded in the cytoplasm. So with all these structures you can see the structure of animal and plant cell. So this is the cytosol. So these are mitochondria. Let us label the parts first. Then these are lysosomes. These are peroxisomes. These are Golgi body, ribosomes, this is a pinocytic vesicle, these are microvilli, these are peroxisomes. Let me write this. Centrioles. So these are vacuoles. So this is RER, that is this is SER, the smooth endoplasmic reticulum, and this is the RER, that is the rough endoplasmic reticulum, and this is the nuclear membrane, the nucleolus, and this is the chromatin material or the chromatin, where the chromosome is in the form of a network which is called as the chromatin. So these are the various structures that can be seen in the animal cell. Here this is vacuole. So this is mitochondria. Plasma membrane. So this is RER, rough endoplasmic reticulum, microtubule. So these are peroxisomes. These are lysosomes. The chloroplast. the Golgi complex or the Golgi body, Golgi complex, Plex. Here also Golgi body. Then this is the cell membrane, cell wall. This is plasma desmata, and this is the plasma membrane. So these are the various organelles of the eukaryotic cells. So you can see the ultrastructure of an animal cell. So the outermost layer is the plasma membrane. So here you can see the plasma membrane has got an infolding forming a rounded structure or a vesicle-like structure, which is called as the pinocytic vesicle, and these are the outgrowths from the cell coming out, which you call them as the microvilli. They increase the surface area of the plasma membrane, and you have a nucleus at the center. So nucleus with nuclear slit, nuclear membrane with slits, and the chromatin or the genetic material inside, and the nucleolus. So the nucleus is also surrounded by the rough endoplasmic reticulum. So this is actually the endoplasmic reticulum where the ribosomes are studded with, and therefore it is called, since the surface forms becomes rough because of the presence of the ribosomes, it is called as the rough endoplasmic reticulum. On the other hand, you also have the smooth endoplasmic reticulum where they are not studded with ribosomes. So there are mitochondria, the double membranous structures. There are centrioles, which are unique for only the animal cells, and they are absent in the plant cells, and these are tiny vacuoles. And you can see the cytosol is nothing but the cytoplasm, the mitochondria, the lysosomes, then peroxisomes. They are the structures which bring about the oxidation in the cells. So plasma membrane, the Golgi body. So this cisternae or plate-like structures, what you can see along with the vesicles, so they are called as the Golgi body. The ribosomes, which help in the synthesis of protein, and these are pinocytic vesicles. So this is the detailed structure of the animal cell as revealed in the electron microscope. So this is the plant cell. So you can see the outermost is the cell wall, and inner to the cell wall is the plasma membrane. So here the cell wall, we have tiny slits in between, which is called as the plasma desmata, which creates the intercellular connections, or it helps in the communication between the adjacent cells. And the plasma membrane can be seen inner to it, and inner to it is the cytoplasm, wherein the so many organelles are embedded in the cytoplasm. Here the central space is occupied by the vacuole, and the membrane is called as, the outermost membrane of the vacuole is called as the tonoplast, and this is the nucleus, which is at the peripheral region and not at the center. So here in animal cell you can see the nucleus is present at the center, but here it is at the peripheral region, and the most of the space in the plant cell is occupied by the vacuole. So there are mitochondria, that is the powerhouse of the cell. Then there are structures like Golgi complex, then rough endoplasmic reticulum, the smooth endoplasmic reticulum, the lysosomes, peroxisomes, microtubules. All these are structures of the plant cell. So chloroplast is unique only to the plant cell. Cell wall is also unique to the plant cell, which are absent in case of animal cells. So this is the ultrastructure of plant and animal. Now we are supposed to understand each and every organelle in detail. We are supposed to study the cell wall, we are supposed to study the plasma membrane, we are supposed to study all the endomembrane system. So the membranous structures which are present inside the cell, all of them we are supposed to study in detail along with the structure and also the functions. So that is the aim of this chapter in particular. So to start with, we will start with the plasma membrane. So these are the eukaryotic cells. So with a very well defined nuclear membrane surrounding the nuclear material. So there is a definite nucleus which is absent in case of prokaryotic cells. Now coming to the plasma membrane. So plasma membrane, as we all know, it forms the outermost boundary of the animal cells and it is enclosed by the cell wall in case of plant cells. So coming to the next is the plasma membrane. Plasma membrane, also called as cell membrane, also called as cell membrane, then also called as plasma lemma, also called as plasma lemma. So this plasma membrane is selectively permeable, thin, elastic, regenerative membrane which surrounds the cytoplasm of all the cells. And it is the outermost membrane or it forms the boundary of the animal cell and it encloses the cell wall in case of, it is enclosed by the cell wall in case of plant cells. So they are covered by the what is called as the cell wall covers the plasma membrane in case of plant cells. And it was discovered by a scientist called as William Seyfried. Was a scientist who discovered the plasma membrane. Now, how is the structure of the plasma membrane? What makes the plasma membrane? What is the chemical composition of this plasma membrane? So the chemical composition of the plasma membrane is, it is made up of carbohydrates, it is made up of proteins, and it is made up of phospholipids. So I hope you all know what are carbohydrates, proteins and the phospholipids. So they are all biological molecules which are organic molecules present in the body of human beings. The carbohydrates, the proteins and the phospholipids. So the carbohydrates are about 7%, the proteins are about 49% and the phospholipids, they are nearly about 44%. So this is the chemical composition of the plasma membrane. So this percentage may vary from cell to cell. For example, in RBC cells, the proteins may be 52% and the carbohydrates may be 88% and remaining may be the phospholipids. Some around 48 or something like that, 42 or something phospholipids are present. So it varies from one cell to another cell according to their functions. So this is the plasma membrane. Now how is the structure of this plasma membrane? When you enlarge the structure of the cytoplasm and how these molecules are arranged in the plasma membrane. So the structure of this plasma membrane has been well explained with the help of a model which is called as the fluid mosaic model. And this was first explained by the scientists called as Singer and Nicholson were the scientists who explained this model which explains the structure of the plasma membrane. Of course, it contains carbohydrates. These carbohydrates, they combine with proteins and they form glycoproteins, they form glycoproteins and they combine with lipids and they form glycolipids. So these carbohydrates, they are not present directly as sugars. So some sugars are also present. Along with that, they combine with the proteins to form, sugars combine with the proteins to form glycoproteins, and the sugars combine with the lipids to form the glycolipids. So proteins, here there are two types of proteins in the plasma membrane, the intrinsic proteins and the extrinsic proteins. Intrinsic proteins and the extrinsic proteins. So intrinsic proteins are also called as integral proteins, which are present inside the lipid layer, and the extrinsic proteins are the one which are present on the surface or the peripheral region of the plasma membrane. So they are called as the extrinsic proteins and intrinsic proteins. Then this phospholipids, so they have it is a bilayer of phospholipid is present. One portion is the head and the other portion is the tail, and the head is polar and the tail is non-polar, and the head is hydrophilic, whereas the tail portion, which is non-polar, is hydrophobic. So they repel from water, they don't get attracted to the water, that is the meaning of hydrophobic, and hydrophilic is they get attracted to the water, that is the head surface. So the phospholipids has the head and tail two portion, and which forms the phospholipids, and the head portion is polar and it is hydrophilic, the tail portion is non-polar and it is hydrophobic. So with the help of a diagram, we will try to understand the fluid mosaic model of the plasma membrane. So this is the exact composition of plasma membrane, where it contains carbohydrates, proteins and phospholipids. So these are the three important components that are present in the plasma membrane, and with the help of a diagram we will see how they are arranged so that they form the structure of the plasma membrane. And finally we will go to the functions of plasma membrane, and then we will go to the study of cell wall. Now coming to the fluid mosaic model of plasma membrane. So plasma membrane is a thin structure, so you know what is a thin structure, and it is regenerative elastic that it can easily expand, and it is permeable, it is selectively permeable, that it will allow some selected substances to pass through it. That is the meaning of selectively permeable. It is elastic, selectively permeable and regenerative. So it can be generated or it can be reconstructed if it is damaged. That is the meaning of regenerative. So coming to the diagram of the fluid mosaic model. Yeah, this is the fluid mosaic model of, instead of writing beneath, I am writing on the side, fluid mosaic model as explained by Singer and Nicholson. So this is called as the fluid mosaic model of the plasma membrane. So look at the structure of the plasma membrane. It looks so small, 70 angstrom unit it is almost 70 angstrom unit, and when you enlarge it, how it looks, see the lipid bilayer and this or the lipid molecules with head on the outwards and tail in the inner side. So this head is the polar end, and it is hydrophilic. The tail is non-polar and it is hydrophilic. So you can see the proteins which are distributed in between the lipid layers, that is called as intrinsic proteins. They are called as the intrinsic proteins. And some proteins are on the peripheral region. You can see here one and here one. These are on the peripheral region on the surface of the plasma membrane, and they are called as the extrinsic proteins. So there are also longer proteins, that is, which are distributed in both the bilayer of the lipid, and they are called as transmembrane proteins or carrier proteins. They are called as transmembrane proteins or the carrier proteins. So this is the structure of the plasma membrane. You can see the glycolipid and the glycoproteins over there. So this forms the structure of the plasma membrane. And now the head and the tail part. Here what you can see is the intrinsic proteins are looking like glaciers which are floating on the sea. So this is also looking like some mosaic or designs in between the lipid layer, which is almost in the fluid state. So it is almost in the fluid state, and these keep mobilizing in the lipid layer. These mosaic-like structures or the intrinsic proteins which are present between the lipid layer, they show the mobility or they show movement, lateral movement within the bilipid layer, and therefore it can be measured as the fluidity of this particular plasma membrane, and therefore it is called as fluid mosaic model. The fluidity of the lipid layer and the movement of these mosaic-like intrinsic proteins within the plasma membrane, that is within the lipid layer, you call it as fluid, hence it is called as fluid mosaic model. So these are intrinsic proteins present in between the lipid layers and the extrinsic proteins which are present on the peripheral part or the surface of the plasma membrane. So since they look like the mosaic in between the lipids, they are called as mosaic, the word mosaic in the model, and fluid because they make a movement, and this lipid is in the form of fluid and they help in the movement of this intrinsic proteins, hence called as fluid mosaic model. So this fluidity also accounts for the cell growth. This fluidity also brings about the cell growth and also formation of intercellular junctions. It also helps in the formation of intercellular junctions. It also helps in cell division. It also helps in cell division. It also helps in secretion. It also helps in endocytosis. It also helps in endocytosis. So once again, the fluidity of this lipid layer helps in cell growth, brings about the cell growth, and most probably it makes the membrane elastic and helps in cell growth and also in cell division. It also brings about the formation of it helps in the formation of the intercellular junctions and also helps in secretion and also endocytosis. Endocytosis is the movement of certain molecules into the cell. So it promotes the movement of molecules, certain molecules inside the cell, hence the word endocytosis. So that is about the fluid mosaic model of Singer and Nicholson, very important from exam point of view. For board exams, it's very important. So that is about the fluid mosaic model. Now let us learn the functions of the plasma membrane. So plasma membrane, we have already told they are selectively permeable. Functions of the plasma membrane. The very first point I would like to write is, it forms the outermost boundary of, so it is the outermost boundary of animal cell. So since they are selectively permeable, they help in movement of selected substances. They help in movement of selected substances into the cell and out of the cell. Substances. Selective is they will select only the required molecules can move into and out of the cell. Hence, it promotes the movement of selected substances. In certain animals like amoeba, it promotes locomotion or movement of the body. It brings about the movement or the locomotion of the body. So, it helps in the movement of the body in case of amoeba, which are unicellular organisms. So, plasma membrane also promotes pinocytosis, which is also called as cell drinking and phagocytosis which is called as cell eating, and also ephagy, which means to remove the unwanted or the waste. So, pinocytosis, cell drinking, if any molecule is present outside the cell. For example, if this is a cell and there is some substance here, so this will form a extension like this, and it will enclose this, and it will cover completely and bring it into the cell, which is called as the pinocytosis or the cell drinking. So, in case of phagocytosis, it will completely cover this. Solid particles are brought inside the cell. Solid particles in the it means the organic molecules are brought inside the cell, which is called as cell eating. So, water molecules are brought inside, it is pinocytosis. With the help of the pinocytic vesicle, they bring it inside, or cell eating, they bring the organic molecules inside. And ephagy. So, if there is some unwanted waste molecule or substance here, once again a vesicle is formed over here, and through this makes an invagination. The plasma membrane undergoes an invagination, surrounds this unwanted substance, and it pulls it out of the cell, and which you call it as the ephagy. So, with the help of the formation of the pinocytic vesicles, they are put out of the cell. So, that is the significance of this plasma membrane and the pinocytic vesicle, which we had shown in the ultrastructure of the animal cell. So, they bring about pinocytosis, that is cell drinking. They bring about the phagocytosis, that is the cell eating. They also bring about the ephagy, or the removal of unwanted or the waste. They throw it out of the cell. Next is, they bring about diffusion. So, there is diffusion of certain respiratory gases into the cell and out of the cell. So, diffusion of gases in and out of the cell, so takes place, which is called as diffusion, with the help of this plasma membrane. Plasma membrane also brings about the osmosis, that is the movement of water molecules. So, the movement of water molecules takes place from a region of higher concentration to the region of lower concentration. You call that as a movement of water also takes place in the plasma membrane. And there are some molecules which are moving inside the cell, you call it as endocytosis. And some substances, they move outside the cell. From the cell, they move outside the cell, you call it as exocytosis. So, they bring about the endocytosis, they bring about the exocytosis, and they also bring about the movement of substances in and out of the cell by endocytosis and exocytosis. So, diffusion, osmosis, endocytosis, exocytosis, then the movement of selected substances, the movement in case of amoeba, all these are the functions of plasma membrane. So, that is the functions of plasma membrane, very important from exam point of view. Next, we will go to cell wall. Once again, any word which is to be repeated, pinocytosis is cell drinking. So, some water molecules are brought inside some water molecules or any other fluid substances which are required for the cell are brought inside the cell if they are outside, present outside with the help of the pinocytic vesicle, which is called as the cell drinking. And phagocytosis, that is cell eating, organic molecules which are present outside the cell are brought inside the cell by the formation of phagosomes, and you call it as phagocytosis. The same phagosomes are put outside, they enclose the unwanted substance and they are put outside the cell, you call it as ephagy. So diffusion, osmosis, almost you people know very well, and endocytosis is movement of molecules inside the cell. Endo always stands for inward movement and cytos always stands for the cell. Exo outside and cyto cell. So movement of molecule into the cell, endocytosis. Movement of molecules outside the cell, you call it as exocytosis. So these are the functions of the plasma membrane. Along with that, I had told the fluid nature of the lipid also brings about cell division, cell growth, then brings about the intercellular formation of intercellular junctions, secretion. All these are also you can add to the functions of the plasma membrane. So thereby we will conclude plasma membrane. We will go to cell wall, study of cell wall. So the cell wall is rigid, non-living, semi-transparent, wall or membrane which forms the outermost covering of plant cells, plant algal cells and fungi. So cell wall are absent only in case of animal cells, but they are present in plant cells, algal cells and in fungi also. So in fungi they are made up of, in case of fungi the cell wall is made up of a substance called as the chitin. In algae they are made up of mannans, galactans and also cellulose, whereas in case of plants they are made up of cellulose, hemicellulose, pectins and also proteins. So the cell wall is rigid, non-living, semi-transparent and forms the outermost boundary of the plant cells, algal cells and also the fungal cells. So in case of fungi the cell wall is made up of chitin. The algae they are made up of mannans, galactans and cellulose, and in plants they are made up of cellulose, hemicellulose, pectins and also proteins. So when we come to the look into how the cell wall is formed. So when you look into the cell wall of an young plant, so a primary wall is formed. A primary wall is formed in the young plant. In the young plant cell, a primary wall is formed which is made up of cellulose and it is thin. Primary wall is formed which is very thin. As the plant grows, as the growth goes on and the cell gets matured, this primary wall on its inner side is replaced by a secondary wall. So this primary wall goes on diminishing and the secondary wall is formed which is multi-layered and it is formed by the deposition of suberin, pectin, lignin, etc. So this forms the secondary wall. So in between the primary and the secondary wall is the middle lamella, which is the middle lamellae, which is formed by calcium and magnesium pectate, which is formed by calcium and magnesium pectate. So, the primary wall is thin, which diminishes as the cell grows, and a secondary wall is formed inner to the primary wall, and it is multi-layered, and it is formed by the deposition of suberin, pectin and lignin. And in between there is middle lamellae, which is made up of calcium and magnesium pectate, which acts like a glue or gum. It acts like a glue or gum so that it gets stick to the adjacent cells. And both this cell wall and the lamellae, they are cut or they have traversed in the middle so that some cytoplasmic connections are formed between the adjacent cells, and which are called as plasmadesmata, So, intercellular cytoplasmic projections or cytoplasmic connections are formed, and these are called as plasmadesmata. So, both the lamellae and the primary wall or the cell wall is transversed so that there is formation of the plasmadesmata, thereby there is cytoplasmic streaming in between the adjacent cells or the neighboring cells. So, primary wall, secondary wall. Primary wall is thin and diminishes slowly as the cell matures and brings about the formation of the secondary wall on its inner side, which is multi-layered, and it is formed by suberin, pectin and lignin. And the middle lamella is made up of calcium and magnesium pectate, which acts like a glue and helps in the attachment of the cells or brings about the closing of the cells, closure of the cells. So, we will go to the functions of the cell wall. We will go to the functions of the cell wall. We are going to the functions of cell wall. Firstly, it provides a definite shape to cells. So, it is providing a definite shape. It forms the outermost boundary and it provides a definite shape to the cell. Secondly, it is providing protection. It is giving protection. That is, it prevents mechanical damage. It prevents mechanical damage or infections so that none of the microorganisms can penetrate into the cell so easily. So, it acts as a barrier. And thirdly, they have the plasmadesmata. Which brings about cytoplasmic connections between the adjacent cells. Cytoplasmic connections between the adjacent cells. And finally, in case of the cell wall in the root hairs, they help in absorption of water and salts. So, these are the functions of the cell wall. Giving a definite shape to the cell, providing protection against mechanical damages and any infections, it acts as a barrier, and the plasmadesmata helps promote cytoplasmic connections between the adjacent cells. And the root hairs, even the cell wall brings about the absorption of water and salts. So, these are some of the functions of the cell wall. So, with this, we shall be concluding this video. Thank you.